Welcome back to the deep dive. Today, we are strapping in for what is probably one of the most fundamental deep dives we've ever done. We are going straight into the nucleus, you could say, of modern medical understanding.
We really are.
Clinical genomics. And if you're a first or second year medical student right now, wrestling with these foundational concepts, this session is absolutely for you.
Yeah, that's so right. I mean, the pace of change in this field has just been explosive. We've gone from the meticulous, very slow process of sequencing one gene at a time,
right,
to using this massive parallel technology that can sequence an entire exome or a whole genome pretty much overnight.
It's completely transformed medicine. I mean, we're talking about a shift from just diagnosing to actually predicting.
Yeah.
We're finding the root causes of these incredibly rare neurodevelopmental diseases. We're directing cancer therapies with precision.
And we're right on the cusp of using this for diagnosis and maybe more importantly, prevention of really common diseases, like diabetes or heart failure.
So our mission today is to really solidify those basic components, the genetic blueprint.
We're going to anchor the key principles, the structure, the regulation, the variations, the inheritance patterns. And crucially, we're going to spend a good amount of time on the new technologies and, the ethics. How do we apply all this data safely for patients?
Exactly. We want these concepts to stick, not just for an exam, but for your entire clinical career.
Let's do it.
Okay, so let's unpack this blueprint. We have to start where the code actually lives.
Right.
The core concept is that genes, these functional units, they're made of DNA, and they're packaged in this really specific hierarchical way inside the cell nucleus.
And that DNA it's the masterpiece, isn't it? It's this long linear sequence of just 4 bases: adenine, cytosine, thymine, and guanine, ACTG.
It's an enormous text file.
It is. 3.1 billion base pairs of material in the human genome. If you stretched it out, it'd be about 2 meters of linear material.
All packed into a space you can't even see. And the structure of that DNA is our first big anchor. Everyone knows it, but the mechanism is so key. We're calling it the twisted ladder, the double helix.
Yep. The sides of the ladder are made of sugar and phosphate. That's the backbone.
And the rungs are the base pairs.
And what medical students have to just burn into their memory is the complementarity.
Right. A only pairs with T.
Using 2 hydrogen bonds. And C only pairs with G.
3 hydrogen bonds.
That specific pairing is the physical and chemical basis for everything. For DNA replication, for all the diagnostic tech we use. If you know the sequence of one strand.
You automatically know the sequence of the other. It's a perfect mirror.
Which is why PCR works, why Sanger sequencing works, why NGS alignment works. It all hinges on that complementarity.
Okay, so back to the 2-meter thread. How do you cram 3.1 billion base pairs into a nucleus that's only a few micrometers wide?
That's the packaging problem.
This is where our next anchor comes in, the spooling system. The DNA thread is wound tightly around these little proteins.
They're called histones. And the basic unit of this packaging is something called the nucleosome.
The nucleosome.
Yeah, and you can literally think of it like thread being wrapped around a bead.
So a bit of thread, a bit of DNA.
About 147 base pairs of DNA, to be precise, wound around a core of 8 histone proteins. This one step compacts the DNA by a factor of about 6.
So we start with the helix, spool it into nucleosomes, and then these strings of nucleosomes form a higher order structure.
Which is chromatin. And the state of this chromatin is, and I can't stress this enough, absolutely crucial.
Why is that?
Because it determines whether a gene is physically accessible to the cell's machinery or not. It's the first major switch for gene expression.
Okay, so we classify this chromatin into 2 states. The first is heterochromatin.
Heterochromatin. This is highly condensed, tightly wound, densely packed. So our anchor for this is that this genetic information is hard to get to.
Hard to get to. So it's transcriptionally.
Silent. The genes in that region are effectively switched off. The machinery just can't get in there to read them.
And the opposite is euchromatin.
Right. Euchromatin is the open, less tightly wound, more relaxed version. So we anchor this as the information that is easy to use.
Because it's open and accessible.
Exactly. The enzymes, the polymerases, all the transcription factors can get in there, bind, and read the sequence. It's active.
And then finally, when the cell is about to divide, all this chromatin gets even more condensed and bundled up into the structures we can actually see under a microscope.
Chromosomes. Right. Humans are diploid organisms, so we have 2 copies of the genome, one from each parent.
Visible as 23 pairs of chromosomes. This complete set is the karyotype.
You've got 22 pairs of autosomes, numbered 1 to 22, and then the 23rd pair, the sex chromosomes.
And just for a quick refresher on the notation you'll see in clinical records, a normal female is 46XX.
And a normal male is 46XY.
Understanding that hierarchy DNA to nucleosome to chromatin to chromosome, it's the foundation for everything that follows.
It is. If you miss this, you miss how the cell controls itself.
Okay, so let's pause for a quick check-in right here. If a cell is highly specialized, let's say a neuron, it's fully differentiated, it never needs to divide again, and it only needs to express maybe 10% of its genes to do its job. What can you logically conclude about the state of the other 90% of its genome?
Well, if that other 90% is not needed for the cell's function, it's going to be packed away.
Right.
It'll be tightly coiled up as heterochromatin, that hard-to-get-to state. This keeps it stable and prevents any wasteful or unwanted gene expression. If you couldn't quite make that leap, just take a moment, rewind, and re-listen to the spooling system and those two chromatin states.
Perfect. So now we move from the static blueprint to the dynamic machinery that actually reads it.
From DNA to protein.
This is the pathway often called the central dogma.
It is. The basic steps are transcription, then splicing, then translation, and finally protein modification. But we have to say, right up front, that this dogma is a bit of an oversimplification.
Why?
Because we have over 20,000 genes, but a single gene can produce many different proteins. And a lot of the genome that regulates this doesn't code for protein at all. The real complexity is in the regulation.
Okay, so let's start with transcription. DNA to RNA. The enzyme doing the work is RNA polymerase.
Right, and it needs to know where to start. That starting line is a region of DNA called the promoter.
And that sits just upstream of the gene's coding sequence.
The RNA polymerase then moves along one of the DNA strands, using it as a template, and it builds a complementary messenger RNA, or mRNA, molecule.
But it uses uracil, U, instead of thymine.
T, correct. And then it stops when it hits a termination signal at the end of the gene.
So let's just nail the differences between RNA and DNA again. RNA is temporary, single-stranded, uses ribose sugar, and has that uracil.
And DNA is stable, double-stranded, uses deoxyribose, and has thymine. Those differences are key. RNA is designed to be temporary, which is why the cell can degrade it so easily.
So what decides if and how fast that RNA polymerase actually works?
That's where the regulation gets incredibly sophisticated. It's not just about the promoter. It's controlled by a huge number of proteins called transcription factors.
Over 1,200 different types in humans?
Yeah, and they can bind right near the promoter, or, and this is a really powerful part, they can bind to distant DNA elements called enhancers.
And when you say distant?
I mean thousands, even hundreds of thousands of base pairs away from the gene they actually regulate.
How does that even work?
It's amazing. The chromosomal DNA literally forms a physical loop which brings that distant enhancer element with all its bound transcription factors into direct physical contact with the RNA polymerase machinery sitting at the promoter.
Wow. So it's like a remote control switch.
Exactly. And that interaction dictates whether that gene is switched on or off.
And this isn't just a theoretical concept, right? This is clinically relevant.
Absolutely. A classic example is a form of alpha thalassemia. Sometimes it's caused by a big deletion of the gene, sure. But it can also be caused by a tiny single base substitution in an enhancer element that's located over 100,000 base pairs away from the alpha globin gene promoter.
So the gene itself is completely fine.
Perfect. But the remote switch, the enhancer, is broken. So you get severely reduced transcription and the patient gets anemia. The machinery is fine, but the switchboard is busted.
Which is the perfect lead-in to our next big anchor, epigenetics. These are the modifications above the DNA sequence that control gene activity.
Yep. They're what fundamentally decide if the chromatin is easy to use or hard to get to. Our anchor here is that epigenetics is the switchboard. It controls the whole flow of information.
And there are two main mechanisms we need to know. First up, DNA methylation.
Right. This involves adding a tiny chemical flag, a methyl group, to cytosine molecules. This usually happens in promoter regions.
And when promoter gets heavily methylated, what's the consequence?
Transcriptional silencing. It's switched off. The methyl groups physically get in the way. They block transcription factors from binding, and they recruit proteins that compact the chromatin, effectively locking that gene into the hard-to-get-to heterochromatin state.
And the second mechanism is histone modification.
Right. Since the DNA is spooled around histones, chemically modifying the tails of those histone proteins changes how tightly the DNA is packed.
So things like methylation or acetylation of the histones.
Exactly. And a good rule of thumb is that histone acetylation tends to loosen the spool of thread. It promotes that open, easy-to-use euchromatin state,
Making the gene active.
while histone methylation can be more complex, but it often contributes to compacting the structure into hard-to-get-to heterochromatin.
From a clinical standpoint, where do we see this go wrong?
Cancer. Abnormal epigenetic patterns are fundamental drivers of cancer. Genes that are supposed to suppress tumors often become hypermethylated
And silenced.
and silenced, allowing the cell to grow out of control.
And this gives us a therapeutic anchor, right? We can target the switchboard itself.
We can. A great example is the drug vorinostat. It's a histone dealotase inhibitor used for cutaneous T-cell lymphoma.
So it inhibits the enzyme that removes acetyl groups.
Exactly. So by blocking their removal, it forces the chromatin to stay in a more open active state. This re-expresses previously silenced tumor suppressor genes, and that can cause the malignant cells to regress. We're treating the control system, not just the mutation.
Okay, so once we have that initial RNA transcript, it's still not ready to go.
Nope, it's still raw. It has to be processed through splicing.
And this is where the cell cuts out the introns, the non-coding bits, and joins together the exons, the coding regions.
Right, and this is done by a giant molecular machine called the spliceosome.
And the genius of this process gives us the alternative splicing anchor.
This is such a cool concept. One gene can produce multiple, totally different proteins with distinct functions.
Oh.
It all depends on which exons are included or excluded in the final mRNA product in a particular cell type.
The classic example being the calcitonin gene.
It's a beautiful one. In your thyroid C cells, splicing produces calcitonin, the hormone that inhibits bone breakdown.
Right.
But in your neurons, the exact same RNA transcript is spliced differently to produce CGRP, calcitonin gene-related peptide.
Which is a neurotransmitter involved in migraines.
Two completely different proteins from one gene just based on how it's spliced.
Amazing. Okay, so once the mature mRNA is made, it leaves the nucleus and goes to the ribosomes for
translation, this is the assembly line where the protein is actually built.
the mRNA sequence is read in triplets, which we call codons.
Yep. There are 64 possible codons. 61 of them code for the 20 amino acids. And the other 3 are stop codons that tell the ribosome to terminate the chain.
And the adapters that bring in the right amino acid are the transfer RNA molecules, the tRNAs.
Correct. Each tRNA has an anticodon that matches a specific codon on the mRNA, ensuring the right amino acid gets added to the growing protein chain.
But even after translation, the protein's not done yet.
No, it has to be folded correctly and often modified. This happens in the ER, the endoplasmic reticulum, and the Golgi apparatus.
Things like adding sugar groups, which is glycosylation.
Right. And this brings us to another huge clinical anchor. The most common variant in cystic fibrosis, which is called delta 500-508 CFTR.
This is a deletion of just one amino acid.
One amino acid, phenylalanine, at position 508. And this tiny change causes the CFTR protein to just slightly misfold.
And that's the key.
That's the key. The cell's quality control system in the ER recognizes this misfolding. And instead of letting the protein travel to the cell membrane to do its job as a chloride channel,
It traps it.
it traps it in the ER and marks it for destruction. So the protein is made, but it never reaches its destination. It's a trafficking problem caused by a quality control failure.
We can't leave this section without mentioning the parts of the genome that don't become proteins but are still functional.
The non-coding RNAs.
Right. We've talked about tRNA and RNA, but the ones that have really exploded in importance are the microRNAs or miRNAs.
We have over 1,000 different types, and they are incredibly powerful regulators.
How do they work?
They're short RNA molecules that bind to complementary sequences on target mRNAs. This binding event usually leads to the degradation of that mRNA, effectively silencing the gene post-transcriptionally.
And they affect what? Over half of all human genes?
And that's the estimate. They are critical for development, for differentiation, and they're heavily implicated in cancer and degenerative diseases.
So if we pull all of that together, what's our first major insight here?
I think it has to be this.
Key insight 1, the machinery principle. The central dogma is this dynamic, heavily regulated process. It's not a simple straight line. You've got the epigenetic switchboard controlling what's even accessible, and then you have non-coding RNAs like miRNAs fine-tuning the output.
And this incredible regulatory system is what allows a fixed number of genes, just over 20,000, to produce the massive diversity of cells and proteins that make up a complex human being.
Right. So let's move on to the life cycle of the cell itself.
Cell division and cell death. This is absolutely vital for understanding inheritance and especially for understanding cancer.
Let's start with division in our normal body cells, our somatic cells. That's mitosis.
Yep. The goal of mitosis is to produce two genetically identical daughter cells, both diploid, both with 46 chromosomes. It's for growth and repair.
And mitosis is preceded by interphase,
Which has three key stages. G1, which is growth, S for synthesis, where the DNA is actually replicated, and G2, which is more growth and a critical last-minute check for errors.
And this whole process is controlled by cell cycle checkpoints.
They're like gates. The cell can't proceed unless everything is correct. The G2 checkpoint, for example, is probably the most clinically important.
Why is that?
It's the last chance a cell has to make sure all the DNA damage has been repaired before it commits to dividing. If that checkpoint fails and the proteins that run it are mutated,
That damaged DNA gets passed on to the daughter cells.
which is a fundamental step in how cancer starts.
So, in sharp contrast to mitosis, we have meiosis.
Meiosis is the specialized two-stage division for making gametes, sperm, and eggs, the germline.
And it has two big goals. First is reduction.
Right, reducing the chromosome number from diploid 46 down to haploid 23.
So that when sperm and egg fuse, the baby has the right number, 46.
Exactly. And this second big goal is recombination.
Also known as crossing over. This is the physical swapping of genetic material between the homologous pairs of chromosomes.
And this happens at a very specific time. Prophase I of meiosis is I.
And this is a massive source of genetic diversity, isn't it?
Huge. The chromosome you pass on to your child isn't just a perfect copy of the one you got from your mom or your dad. It's a hybrid, a mix of the two. It's why siblings from the same parents are all different.
Now, the timing of meiosis is really different between the sexes, and that's clinically important.
Vitally important. In males, meiosis starts at puberty, it's continuous, and it completes in a matter of days.
But in females,
In females, meiosis the feared begins when they are a fetus, and then it pauses. It stops in prophase 1. Meiosis II only completes if and when that egg is ovulated and fertilized, which could be 40 years later.
and that long pause is the clinical anchor for one of the most common genetic problems we see.
The increased risk of trisomies, like Down syndrome, with advanced maternal age.
Why does that happen?
It's because that meiotic machinery, which has been paused for decades, becomes less efficient at separating the chromosomes correctly.
This error is called non-disjunction.
Right. And if it happens in meiosis II, you end up with two normal gametes and two abnormal ones. But if non-disjunction happens back in meiosis the set, all four resulting gametes will be abnormal. It's a major source of aneuploidy.
Okay, moving from cell replication to cell death. There's more than one way for a cell to die.
Right. And not all of them are bad.
The first fate is senescence.
Senescence is when a cell just stops dividing. It hits the Hayflick limit, the point where its telomeres, the protective caps on the chromosomes, get too short.
And this is part of normal aging. But when it goes wrong, you can get premature aging syndrome.
Exactly. Our clinical anchor here is Werner syndrome. It's caused by a mutation in a DNA helicase, which is an enzyme needed for DNA repair. So the cells can't repair damage properly, they hit their Hayflick limit way too early, and the person ages prematurely.
Then we have apoptosis, programmed cell death.
This is an active, highly controlled process. It's essential for development like getting rid of the webbing between our fingers and toes as an embryo.
And also for the immune system, right? Killing off infected cells.
Exactly. The process is triggered by signals that activate enzymes called caspases. These are the molecular executioners. They chop up the cell's components in a very orderly way.
And the cell fragments into these little membrane-bound sacs called apoptotic bodies.
And that's the key. Macrophages can then come along and just gobble them up. It's a clean, quiet, non-inflammatory process.
Which is a huge contrast to necrosis.
Necrosis is pathological. It's uncontrolled cell death. It happens from a major injury, most commonly hypoxia, lack of oxygen.
So the cell membrane breaks down.
It ruptures. The cell contents spill out everywhere, and that triggers a big, damaging, inflammatory response. It's a messy, chaotic death.
Okay, let's do another quick check-in. We talked about recombination as being this huge source of diversity. Let's focus on that non-disjunction error again. Can you recall the difference in outcome if non-disjunction happens in meiosis of I versus meiosis II? Why is the meiosis I error more severe?
Well, if the error is in meiosis II, the homologous chromosomes fail to separate. So every single one of the four resulting gametes is abnormal.
Right.
But if the error is later, in the meiosis II, the sister chromatids fail to separate. In that case, you get two normal gametes and two abnormal ones. So the error in meiosis high affects the entire output. If you struggle with that, rewind and really focus on the two stages of meiosis.
Okay, so now that we understand the cell, let's talk about variations in the genetic code itself, the classes and consequences.
We have to start by saying variation is normal. It's what makes us different. Each of us has 3 to 5 million variants compared to the reference genome.
And most of them are harmless polymorphisms. The most common type is the single nucleotide polymorphism, or SNP, a single base change.
But when that substitution happens inside a protein coding region, we have to classify its effect. We can use the analogy of reading a sentence.
First up, the synonymous or silent substitution.
A base changes, but because of that codon redundancy we talked about, it still codes for the exact same amino acid.
So our analogy is changing cat to cat, the meaning is exactly the same.
Right. Then there's the non-synonymous or missense substitution. A base change leads to a different codon, which specifies a different amino acid.
Our analogy, changing FOX to BOX, the meaning is lost or fundamentally changed.
And the severity of that depends entirely on where it is in the protein and what kind of amino acid change it is.
Then you have the really dramatic one, the stop-gain or nonsense substitution.
This is where the change creates a premature stop codon. The translation machinery just stops, and you get a truncated, usually non-functional protein.
The analogy, F8 becomes cat, and the sentence just ends abruptly. The rest of the information is gone.
And we can't forget splicing variants. These are substitutions right at the intronexin boundaries. They mess up the signals for the spliceosome, leading to exons being skipped or introns being left in.
Which usually leads to a non-functional protein.
Almost always.
Okay, moving beyond single base changes, we have insertions and deletions, or indels.
Right. If you insert or delete a multiple of 3 bases, 3, 6, 9, the reading frame downstream stays intact. We call that an in-frame indel.
So the protein might be missing an amino acid, but the rest of it is correct.
But if you insert or delete 1 or 2 bases, that's a frame shift variant.
And this is catastrophic.
Totally catastrophic. It shifts the entire triplet reading frame for every single codon that comes after it. The whole downstream sequence of amino acids is just garbage, and you usually hit a stop codon pretty quickly. It's almost always pathogenic.
Next, we have length variations, simple tandem repeats.
These are short DNA sequences that are repeated over and over again, and they're very unstable. They can expand or contract during replication.
And this instability is the basis for the triplet repeat diseases.
This is a critical clinical anchor. You have these unstable repeats, like the Chi gene repeat in Huntington's disease.
And these repeats can actually expand from one generation to the next.
A process called anticipation. The disease appears earlier and gets more severe in subsequent generations. It's a very unique pattern of inheritance.
Scaling up again, we have copy number variations, or CNVs, gains or losses of big chunks of DNA up to whole chromosomes.
And the whole chromosome gains, trisomies, are usually caused by that meiotic error we just discussed, non-disjunction.
Trisomy 21 is Down syndrome, 18 is Edwards, 13 is Patau.
And whole chromosome losses, monosomies, are usually lethal, with the exception of sex chromosome monosomies like Turner syndrome, which is 45X.
And finally, the really large structural anomalies.
These usually happen when DNA breaks and is then repaired incorrectly. One mechanism is non-homologous end joining. The cell just glues 2 broken ends together, even if they don't belong together.
Which can lead to a translocation where a piece of one chromosome gets stuck onto another.
Exactly, or an inversion or a deletion. A second, more insidious mechanism is non-allelic homologous recombination, or NAHR.
This sounds complicated.
It's an error caused by the machinery misaligning on regions of the genome that have similar repeated sequences. It tries to recombine them, but because they're misaligned, it swaps unequal segments.
And this causes the recurrent microdeletion syndromes.
Precisely. This is why some syndromes, like the 22q11.2 deletion syndrome or DiGeorge syndrome, are relatively common. That region is a hotspot for this kind of NAHR error during meiosis.
Okay, so once we find a variant, we have to figure out its functional consequence.
Right. What is it actually doing to the protein?
The classic effect is loss of function.
Reduced or completely absent protein function. This is what you get from a gene deletion, or an early nonsense or frameshift variant. This is often the basis for recessive diseases, because one working copy is usually enough.
Then you have gain of function.
This is where the variant gives the protein a new or altered function. For example, a missense variant might make an enzyme permanently switched on, driving unregulated cell growth. This often leads to dominant diseases.
And the third type, which is a bit tricky, is the dominant negative effect.
This happens when the abnormal protein produced from one allele actually interferes with the function of the normal protein from the other allele. It actively poisons the system.
A great example is in osteogenesis imperfecta, brittle bone disease.
Perfect example. The mutant collagen protein gets incorporated into the collagen fiber, but makes the whole structure unstable and weak. It's a much more severe disease than if you just had a simple loss of one copy.
So understanding the functional consequence is key to predicting the disease.
It's the whole game.
So this gives us our second key insight.
Key insight 2, the predictability of variants. Genetic variation is normal, but pathogenic variants cause either a loss of function or gain or interference of function. And knowing which one it is, which you can often infer from the type of variant, helps you predict the inheritance pattern, the severity, and what kind of drug might even work.
Right. Now let's map these variants onto families and inheritance patterns, starting with constitutional genetic disease.
This is what's inherited through the germline. And our first tool, our fundamental tool, is the pedigree analysis.
A 3-generation family tree. Squares for males, circles for females, shaded if they're affected. It's the visual map.
Let's start with autosomal dominant, or AD, inheritance. The key feature here is you see affected people in every generation.
Unless it's a de novo variant, a new mutation.
Right. You only need one copy of the pathogenic variant to have the disease, and the transmission risk to each child is 50-50.
And with AD conditions, there are 2 terms you have to master: penetrance and expressivity.
Penetrance is the proportion of people with the variant who actually show the disease. If a variant is 80% penetrant, 20% of people who have it will be perfectly healthy.
We see that a lot in cancer predisposition, like with BRCA12.
And expressivity is the range of severity. In Marfan syndrome, 2 people with the exact same variant can have wildly different symptoms. One might just be tall, the other could have a life-threatening aortic dissection. That's variable expressivity.
Next, autosomal recessive, or AR inheritance.
For AR, you need 2 copies of the mutated allele for the disease to show up. The parents are usually healthy carriers.
So it often appears out of the blue in a single generation.
Exactly. And if both parents are carriers, the risk for each child is 25% affected.
And you see AR conditions more frequently if the parents are blood relatives or consanguineous.
Because they're more likely to share the same rare mutant allele from a common ancestor. Cystic fibrosis, sickle cell disease, these are classic AR conditions.
The third Meiji pattern is X-linked inheritance,
which mostly affects males because they only have one X chromosome. They're homozygous.
An affected male can't pass it to his sons because he gives them his Y chromosome.
But all of his daughters will get his X, making them obligate carriers. So transmission is typically from a carrier mother to her son, with a 50% risk for each son.
But sometimes female carriers can show mild symptoms. Why is that?
This is due to skewed X inactivation. Early in development, in every female cell, one of the 2 X chromosomes is randomly switched off.
So it's a 50-50 chance which one gets inactivated?
Usually. But if, just by chance, in a lot of the key tissues, it's the normal X chromosome that gets inactivated, then the mutant allele on the active X gets expressed, and she can show mild symptoms.
Okay, and the last pattern is mitochondrial inheritance.
This is exclusively maternal inheritance. All our mitochondria come from our mother's egg cell.
So it affects males and females equally, but an affected male cannot pass it on.
Correct. And the key feature here is massive variability, which is due to mtDNA heteroplasmy. Each cell has hundreds of mitochondria, and you can have a mix of normal and mutated mitochondrial DNA within a single cell. Disease only happens when the percentage of mutated mtDNA crosses a certain threshold in a particular tissue.
And since mitochondria are all about energy production,
the defects hit the tissues with the highest energy demand the hardest: muscle, the retina, parts of the brain.
Okay, our final pattern is somatic genetic disease.
These are variants that are not inherited. They happen after fertilization during mitotic divisions in body cells.
And the most important example is cancer.
Right. Cancer is driven by somatic mutations in oncogenes, which promote growth, or tumor suppressor genes, which regulate growth.
And this brings us to the Knudsen 2-hit hypothesis.
In a sporadic cancer, you need 2 random unlucky somatic hits to knock out both copies of a tumor suppressor gene in a single cell.
But in a familial cancer syndrome, the person inherits the first hit. It's already in every cell of their body.
So they only need one more random somatic hit to lose the protection from that gene, which is why their lifetime risk of cancer is so much higher, and it happens at a younger age.
That distinction between constitutional and somatic is just paramount.
Absolutely.
Okay, quick check-in on X-linked inheritance. A woman is a known carrier for a serious X-linked condition. She's pregnant. What's the fundamental difference in risk for a female fetus compared to a male fetus?
For a male fetus, the risk of being affected is a straight 50%.
Right.
For a female fetus, the risk of being a carrier is 50%, but the risk of being clinically affected is generally very low. That's because of X inactivation. The normal X chromosome in most of our cells will compensate. It's a critical point for genetic counseling.
Okay, let's talk about the tech, the tools we use to interrogate the genome. This is where things have changed so fast.
Incredibly fast. Historically, we use karyotyping. Looking at chromosomes down a microscope, you could see big changes, but nothing smaller than about 4 megabases.
And today, the workhorse for looking at gains and losses of DNA is array comparative genomic hybridization, or array-CGH.
Right. It's replaced karyotyping in most settings because its resolution is so much higher. It can detect tiny microdeletions and microduplications.
Let's re-anchor the mechanism. You take patient DNA and label it green. You take reference DNA and label it red.
And you mix them together and let them compete to bind to spots on an array. If everything is normal, you get a 1-to-1 ratio of red and green, which looks yellow.
If the patient has a deletion, there's less green DNA, so the spot looks red.
And if the patient has a duplication, there's more green DNA, so the stat looks green. It's a beautifully simple concept.
But array-CGH has 2 massive limitations that everyone needs to remember.
Yes. First, it will not detect balanced structural rearrangements like a translocation where 2 chromosomes just swap pieces. No DNA is lost or gained, so the ratio stays 1-to-1. It all looks yellow. And second, it won't detect triploidy, where there's an entire extra set of chromosomes.
Well,
Because again, the overall ratio of patient to reference DNA is the same. It can only detect relative segmental changes.
and the power of arrays has created new interpretive challenges like the CNV of uncertain significance, or VUS.
Right. We find a deletion or duplication, but we just don't have enough data yet to say for sure if it's benign or if it's causing the disease.
And then there are the neurosusceptibility loci.
These are CNVs that are clearly associated with an increased risk for things like autism or schizophrenia, but they aren't deterministic. Most people with the CNV won't get the condition. There have to be other genetic or environmental factors at play.
And of course, incidental findings. Finding something you weren't looking for, like a BRCA1 deletion in a child being tested for intellectual disability.
A huge ethical challenge.
So moving from big structural changes to the single base level, the foundational technology is still the polymerase chain reaction, PCR.
The DNA copy machine. It's how we amplify a tiny amount of DNA into enough material to actually analyze.
And PCR was what enabled Sanger sequencing, which for decades was the gold standard for reading the exact order of bases.
But it was slow and expensive. The Human Genome Project took over a decade and cost billions.
Which is why the invention of next-generation sequencing, or NGS, was such a revolution.
Instead of one reaction, we're doing millions of sequencing reactions all at the same time. Massive parallelism.
And this brought the cost of a whole genome down from billions of dollars to less than 1,000, and the time from years to a single day.
It made genomics scalable for the clinic.
The NGS process has a few key steps. First, library preparation.
Chopping up the DNA and adding adapters. Then amplification, where you make clusters of identical DNA molecules on a flow cell.
Then the optional step of capture, where you decide what you're going to sequence.
And then the actual sequencing, and finally the computational part, alignment, where you map all those millions of short reads back to the reference genome to find the variants.
A critical metric here is coverage, or depth.
Right? How many times did you independently sequence a given base? For diagnostics, you generally want a coverage of at least 30x.
And knowing the 3 main capture strategies is essential for any clinician ordering these tests.
First, you have whole genome sequencing, WGS. You sequence everything. It's the most comprehensive, but also the most expensive, and generates a ton of data, including millions of variants of uncertain significance in the non-coding regions.
Then you have whole exome sequencing, WES.
This is cheaper. You only capture and sequence the exome, the 1% to 2% of the genome that actually codes for protein. It's a great balance of cost and utility, but you'll miss any regulatory variants in the introns.
And finally, gene panels.
The most cost-effective. You only sequence a pre-selected list of genes known to be associated with a specific condition. This gives you very deep coverage, which is great for detecting mosaicism, but you are completely blind to any novel or unexpected genes.
And the tech is still moving. We now have 3rd-generation sequencing.
Like single-molecule real-time or SMRT sequencing. The key advantage here is that it produces very, very long reads, tens of thousands of bases long.
Which is great for resolving complex, repetitive regions of the genome that NGS struggles with.
Exactly. It's likely to be the future.
So if we synthesize this whole technological revolution, what's the key takeaway for a clinician?
It has to be this one.
Key insight 3, choosing the right tool. NGS has taken over, but you have to know which version to use. WGS for breadth, WES for a good general look, and a panel for depth when you have a very specific question. And you can't forget that array CGH is still essential for those structural CMVs, but you have to remember its limitations.
Right. No single test can see everything.
So let's bring this all into the clinic. Real scenarios, starting with rare diseases.
I mean, individually they're rare, but collectively they're common. And for families with a child with a severe neurodevelopmental disorder, NGS has been a complete game changer.
The diagnostic odyssey.
Exactly. The diagnostic yield used to be terrible. Now with WES or WGS, we're getting a diagnosis in 35 to 50% of cases. That provides answers, prognosis, and allows for reproductive planning. It's huge.
And genomics is also moving into common complex diseases.
Yes, through things like polygenic risk scores or PRS. These are developed from huge GWA studies.
And they're designed to estimate an individual's lifetime risk for things like heart disease or diabetes.
The idea is to use them to target screening and preventative measures to the people at highest risk. They're still in development, but they're coming.
In obstetrics, the change is already here.
Non-invasive prenatal testing, NIPT. We're sequencing the cell-free fetal DNA that circulates in the mother's blood.
And it's an incredibly accurate screening test for the common aneuploidies like trisomy 21, Down syndrome.
Over 98% accurate, which is different from NIPD, non-invasive prenatal diagnosis, which is used to look for specific single-gene defects.
But oncology is where the therapeutic impact is most immediate and dramatic.
For sure. Genetic testing is no longer just about future risk. It's about guiding treatment right now.
We have to unpack the mechanism of synthetic lethality using the example of BRCA12 and PRP inhibitors.
Okay, so BRCA1 and BRCA2 are genes that repair double-strand DNA breaks. When a tumor cell has lost both copies of its BRCA gene, it can't do that type of repair.
So it becomes totally dependent on its backup repair system.
A single-strand repair pathway that uses an enzyme called PARP.
So if we give that patient a PRP inhibitor drug,
we block its only remaining DNA repair pathway. The cancer cell now has zero ability to fix its DNA, it accumulates catastrophic damage, and it dies.
And the beauty is that the healthy cells, which still have one working copy of BRCA, are mostly unaffected.
That's synthetic lethality. It's why BRCA testing is now critical for managing patients with ovarian, breast, prostate, and pancreatic cancer. It determines their immediate treatment options.
Genomics is also changing everyday prescribing through pharmacogenomics.
Using a patient's genetic profile to pick the right drug at the right dose.
A key example is testing for variants in the gene TPMT.
TPMT is an enzyme that metabolizes drugs, like azathioprine. If you're a poor metabolizer because you have certain TPMT variants and you get a standard dose,
the drug builds up to toxic levels.
And you get life-threatening bone marrow suppression.
Yeah.
So now we screen for these variants routinely. If a patient is a poor metabolizer, we give them a tiny fraction of the standard dose, and it's perfectly safe.
And looking to the future, we have to talk about gene therapy and editing.
For sure. We've had success with ex vivo gene replacement for some immune deficiencies.
But the tool that has everyone so excited is CRISPR-Cas9.
It's like molecular scissors that we can guide to any exact location in the genome to make a precise cut or edit.
And while editing cells outside the body is becoming a reality,
the holy grail is editing cells inside the body in vivo, but that still faces huge challenges with delivery and safety.
And we shouldn't forget pathway medicine.
Right. Targeting the downstream effects of a mutation, even if you can't fix the mutation itself.
Marfan syndrome is the classic example. The defect is in the fibrillin gene, but a major downstream consequence is overactivation of the TGF-beta signaling pathway, which damages the aorta.
And it turns out that the common blood pressure drug, losartan, also partially blocks that TGF-beta signaling. So clinical trials have shown it can help prevent aortic dilation in Marfan patients.
It's the essence of precision medicine.
It is.
But as the technology gets more powerful, the ethical questions get more profound.
The guiding principle has to be that a genetic result is permanent and has implications for the whole family. Therefore, it requires informed consent from a competent adult after professional counseling. Full stop.
And this is most critical in predictive testing for an unaffected person.
And for adult onset conditions, The core ethical rule is avoid testing children. We have to preserve their future right to decide for themselves if they want that information, especially if there's nothing we can do to prevent the disease in childhood.
Then there's the rise of direct-to-consumer testing.
Which is tricky. It provides access, but it completely bypasses that essential professional support and counseling.
And the risk profiles they provide for common diseases are often based on a limited number of SNPs and can be misleading.
Hugely misleading. It can lead to false reassurance where someone skips screening they should be having, or it can cause immense anxiety over a result that isn't even clinically validated. It's a major problem.
So we've completed a huge tour of clinical genomics. We've anchored the key structures and the cutting-edge applications.
Yep. We established the blueprint with the twisted ladder and the spooling system. We detailed the regulatory machinery with the epigenetic switchboard, clarified the inheritance patterns, and anchored the technology from array CGH to NGS.
And the speed and the falling cost of WES and WGS means this is moving out of specialized genetics clinics and into every single part of medicine.
It's true. As a future clinician, you will increasingly be confronted with unexpected incidental findings. You'll find a BRCA1 deletion in a patient you tested for a neurodevelopmental delay.
So our final thought for you to consider is this. Given our ethical duty to act on information that can benefit a patient, how should we as medical professionals be trained to manage this complex, life-altering, and completely unexpected information?
The challenge isn't can we sequence anymore. It's how do we responsibly manage this flood of non-targeted risk data that we're about to unleash.
That transition from pure diagnostics to managing unexpected lifelong risk is the defining ethical challenge you will face in the genomic era.
We hope these anchors stick with you as you continue your training.
And we'll catch you next time for the next deep dive.